is Michael Ringel. And Michael is a JD and PhD from Harvard and Princeton. Was a director for the Boston Consulting Group, where he has done an awful lot of really important global analysis that I think pointed him in the direction of this aging research field. He was a wonderful speaker that I heard in a number of different conferences giving these general overviews, but from a little bit of a distance. And just recently, he's kind of entered the field a little bit more and has joined a fascinating organization, Life Biosciences. And yeah, I think sometimes when you go to multiple conferences, you wonder what is interesting, what is potential, what is new. And I think this, what Michael is going to talk to us about now, fits all of that category, or those categories. So without further ado, please welcome Michael Ringel.
There's a sizable portion of the clinical staff, on the whole island here in one place. And so the fact that you're all hearing some of these stories together, we'll be going out afterwards talking to each other, there's actually a nucleus of focus on aging biology that I think no other place in the world has actually been able to gather such a large portion of the clinical staff from the entire country in one place. So super excited to be a part of this and share a bit of my journey. If I could get my slides up, and it's not the deck I see right now. The Accelerated Biosciences is the one after me. Oh yeah, this one, yep, perfect. So as Martin said, I was at BCG for a long time, and I did a project for a client scanning the landscape of aging biology potential pathways, and I just fell in love with one in particular that I will tell you about, partial epigenetic reprogramming. It's a little ways from clinical practice, so for those of you purely focused on that, it'll be a little time before it comes, but I think you'll be fascinated to see what could transform medicine, and so much so that I gave up my job and jumped into a startup in this space. There will be some forward-looking statements as part of that, and I'm actually not going to start with it. I'm going to start with two other topics before leading into it, which are, one is for the finance ministers of the world, or those interested in finance, and the other is for the health ministers of the world and those interested in that edge of things. So just how much value there is here, and that actually it's the only way forward for medicine from where we are today. And then I'll talk in the third chapter about what I think is the philosopher's stone here. So why is there so much value here? Just from an economic perspective, forget the moral value, right, of helping people live longer and healthier. But sometimes people worry that would be a problem economically. We're going to have all these old people hanging around, and won't that be a drain on the economic coffers? But nothing could be further from the truth. That's actually 180 degrees wrong. The only answer to the gray wave, which of course everywhere in the world is experiencing, starting with Korea and Japan and parts of the Far East and then Europe, Mauritius among them. But even the places with the youngest demographics right now that will, they're not that far behind. They will eventually have these demographic pyramids that are cylinder shaped, and you need a new way to think about how you support the economy in that base. And the only answer is actually making sure the top of that pyramid is healthy and capable. A lot of serious economists have looked at this, starting with Murphy and Topol at the University of Chicago. They wrote a National Bureau of Economics research paper, which is like a gold standard economics paper, showing that the historical improvements that we had in health span and lifespan just in the US were worth trillions of dollars a year to the economy. Then Dana Goldman in the middle here at USC used a model that was developed at Rand Corporation to model a very small change to the lifespan curve in humans, going from the blue line here to the purple line here. So if you can see the difference there, you have good eyesight. It's a very small change to the survival curve. And moreover, they phased this in very slowly over a 20-year period. But even this minuscule change, they calculated was worth $7 trillion. And then most recently, Andrew Scott at London Business School looked at a variety of scenarios, actually using the same exact model from Murphy and Topol, but rather than looking retrospectively, now looking prospectively. And in a particular example where they said, let's imagine we can extend health span by 10 years, they showed that was worth $367 trillion, which is a staggering number. For most of us, it probably has no meaning. But to help you kind of put that in context, that's between 2 and 3 times the entire GDP of the world in 1 year. So there's unbelievable economic value in realizing this promise of geroscience, and it comes from 3 things. The fact that people add more to GDP because they can work longer if they want, maybe not in every jurisdiction. Obviously retirement ages are somewhat contentious in some places, but some people will want to work longer. And certainly they act as consumers longer, which also drives the economy. Less reduction from GDP because you're delaying and probably also reducing the cost of healthcare and other entitlements. And then, of course, there is some economic value ascribed to life itself. So that's chapter 1. There's incredible value here for the companies that make this possible, and then for the world that obviously benefits from it. Okay, it's the only way forward for medicine. So this chapter is for the health ministers. And the great starting point is this curve, which is a survival curve in humans, also called a Kaplan-Meier curve. So you see it starts at 100%, goes down to 0. And you can see it's plotted for 4 different cohorts. And there's been incredible improvement in the last 150 years in the shape of this curve, largely due to sanitation, vaccination, and reduced infant mortality. But there are 2 big gains that are missing. The first is health span, the quality of those years. We can expect to spend a decade on average in ill health. And the second is, despite the improvements in average lifespan, we haven't changed maximum lifespan. That's true no matter how much you spend. So you saw a version of this chart yesterday in Mahmoud's presentation. So you've got healthcare spending on the x-axis, you've got health-adjusted life expectancy on the y-axis. Each dot is a country. Yes, that dot way out to the far right is the US, but it's not about that specific country. There's obviously a pattern here that it looks like you're running up against a wall. And whenever you see something that looks like that, you should ask the question, well, is there some sort of natural limit that we're running up against? And the answer to that is yes, that is the limit. Jay Olshansky and colleagues wrote a great paper in Science in 1990, showing that if you want to get to an average lifespan of 85, of 90, of 105, you would have to reduce mortality from then current rates by 55, 70, 90%. And that's in the context that even if you cured all major degenerative disease, you would only get a 75% reduction. And so they made the prediction that you see on the right-hand side here that the period of rapid increase in life expectancy has come to an end. They took a ton of criticism for this at the time, but they've been absolutely vindicated. It's flattened out around the world, in the developed world, and in some places actually regressed, largely due to obesity. But this isn't their exact quote. What they actually said was unless we can alter aging at the molecular level. And the reason why that could actually solve the barrier that we're running up against is this chart, the geroscience thesis, that the number 1 risk factor for disease is age. More than 90% of what kills us in the developed world are age-related diseases. You can see they go up with age and exponentially with age. And if you could actually modulate where you are on this x-axis by changing your rate of aging, then you can change the incidence of all major degenerative disease simultaneously, exactly the challenge that Jay Olshansky put to us. So it's the only way to solve the barrier is something that's going to hit all of these things simultaneously. And thankfully, there's a lot of evidence that it's feasible. We have evidence from genetic variants. This is an example from worms. We're looking at a survival curve or a Kaplan-Meier curve again. This is one of the papers that really kicked off the field. Cynthia Kenyon's paper showing DAF2, this single gene mutation, makes the worms live twice as long. So from the white line to the black line. And not only do they live twice as long, but they're healthier all along the way. All their degenerative diseases are moved out, such as worms have degenerative diseases. They're not getting Alzheimer's, but their motility, things like that. We see it in pharmacological interventions. This is an example from mice. There's something called the National Institutes on Aging Interventions Testing Program, ITP, where they test a lot of different small molecules to see if they extend lifespan, and this is the first one that was ever shown to have an effect. This is rapamycin. You can see the mice are living longer, and they're healthier all along the way. Their degenerative diseases, things like cancer, are pushed out. And lastly, we see it in epidemiology. This is an example from humans, looking at different populations of Japanese, from sumo wrestlers to mainland to Okinawans who are in a famous blue zone, and the amount of caloric restriction they practice. Pretty much every blue zone has caloric restriction as the core component. And you can see that there's a relationship. This is the best validated intervention in animals, and it appears to also have an effect in humans. That's the good news. To understand the limits of this, though, I want to talk to you about why this works. It's weird, right? So if you're starving an animal, almost to the brink of killing it, you make it live longer. And many other stresses, you put the animal under stress, more and more stress, longer and longer life, up to a point where then you kill it. That's weird, right? Why would that be so? And there are various explanations out there, but there's only one correct one. I don't have long enough in this presentation. I have a different presentation to explain why it's the right one, but I'm going to just assert it, given time. And the logic is as following. So energy and resources are not infinite. They're limited. So organisms have a choice in how they deploy these limited resources across many different uses, but you can kind of grossly simplify it to growth and reproduction on the one hand, maintenance and repair on the other hand. And if you're in the middle of a famine, like under caloric restriction, offspring are unlikely to survive, certainly to thrive. And so, yes, energy and resources are down, but organisms actually even greater reduce the amount that they put into growth and reproduction and shift a little bit of excess to maintenance and repair. And actually repair damage, continue on, try to wait out the bad period. And this isn't an intentional thing, obviously. This is something that's been selected by evolution. And there's a lot of evidence that this is actually what's going on, including the genes that first discovered showing this, like DAF2, that I showed you, are diapause genes. These are genes actually involved in the pathway that put animals into this quiescent state where they wait out bad periods. And if we think about all the interventions that have ever been shown to work for maximum lifespan in the interventions testing program, we can actually map them on this master switch for plasticity between, hey, if conditions are good, grow and reproduce, and if they're bad, maintain, repair, stretch, wait. So the switch should sense conditions for growth. Are building blocks in place? Is there enough oxygen? Do I have a template suitable for replication? Crucially, do I have enough energy? That switch exists. It's called mTOR. And that switch should then turn on or off actions of growth or repair and maintenance. So on the growth side, protein, lipid, nucleotide synthesis, putting mitochondria, putting energy into a form that's easy to use, and turning off these repair and recycling mechanisms, autophagy and proteasome ubiquitin system, and that's exactly what we see. And if you were then to plot the things that work in the ITP and guess where would the most effective thing sit in this diagram, you would probably guess correctly. It sits right there. The only other two that have ever shown a 10% maximum lifespan effect sit right here. Everything else that's ever worked sits here. There are others that people talk about, spermidine, metformin, NMN. They also fit. And if I've missed out your favorite one, like there's a place for it somewhere on this map. The dirty secret of this, though, if you go back to this switch mechanism, it really matters if you're a short-lived organism. So if you're a worm, and you're waiting for rainfall, an extra week is a big deal. Or if you're a fruit fly and you're waiting for a rotten fruit to fall, an extra month is a big deal. If you already live 80 years, you're averaging out a lot of this variability in conditions. And so the prediction of this is that these mechanisms matter a lot more in short-lived organisms. That prediction is validated empirically. This is a little bit of a hodgepodge chart because it's all different sources and different methodologies, but in general, the trend is going to be correct, where the amount of impact that you get from caloric restriction decreases for longer-lived organisms, so much so that at rhesus monkeys, one study saw a result, the other didn't, because you get to the kind of noise range and sample size. And the estimate in humans is that a lifetime of caloric restriction isn't going to change maximum lifespan a lot. And by the way, we already have seen that epidemiologically, right? Yes, Okinawans are healthier, they do live longer, but they're not living regularly past 120. And so anything that hits this master switch, it's not a bad thing, but it's unlikely to reshape our maximum lifespan. It's still a good thing, don't mishear me on this. You should do exercise and diet and sleep and all the things you hear about, because it's going to greatly reduce your chance of early mortality, and it will increase your health span on average, but it's unlikely to radically shift our maximum lifespan. There is, though, something that taps into a totally different biology. And because it taps into a totally different biology, I believe it has a totally different limit. And there's good evidence now that this is a way forward, which is why I was so excited about it. So what is that biology? It's the biology that makes babies young. So this is actually a picture of my wife when she was 36 and our youngest son, Matthew. Matthew, at one point, was just an old egg cell and a little bit of a contribution from an old male. And that egg cell was old. Right? You could measure it functionally, transcriptionally, epigenetically. It was old. He was old. He obviously wasn't born old, right? He was born completely rejuvenated. And so there has to be a mechanism in nature that takes old cells and makes them young again. Otherwise, babies could not be born young from our old cells. And we actually now know what that mechanism is, which is between day 7 and 9 of embryogenesis, what's called the epigenetic marks, the methyl groups, acetyl groups that are on the DNA, that are on the histones the DNA is wrapped around, are wiped clean. And if you measure the egg cell after fertilization, but before day 7, it will still measure old, functionally, transcriptionally, epigenetically. And if you measure it after day 9, it will measure 0 on an epigenetic clock, and similar to that, not exactly 0, but close to that on transcriptional or functionally. So we already know the thing that in nature can make cells young again, and we've already actually tapped into it artificially. Shin Yamanaka won the Nobel Prize in 2012, showing that you could treat cells with just 4 small proteins and make them into induced pluripotent stem cells. I'm not talking about stem cell therapy, which of course many of you have heard of, but the hidden fact in his discovery was not only did he make those cells pluripotent and that they could then become everything, like a stem cell can become a nerve cell or a muscle cell or whatever, but he also completely rejuvenated those cells. So if you take an old cell, an old skin cell from a mouse, and you treat it with OSKM, and then you grow a clone from it, the clone is born a baby, even though it was just an old skin cell. And in fact, the mouse on the left you see there was an old skin cell that was then born as a baby and grew up and had a normal life, and it's so normal that its pups are shown there on the right. So the thing that people find conceptually hard to believe, that we can actually rejuvenate and completely reverse aging, we've actually already done at the single cell level in the context of making a clone, which I think is incredibly profound, and it's this hidden discovery, right? Hidden fact that exists in the fact that we all know babies are born young, and hidden discovery in this cloning technology. But as you think about it, you realize what it implies for rejuvenation. Now, the problem is you can't do that in vivo, because you don't want to take all your cells back to stem cells. You would cease to function. Your nerve cells have to stay nerve cells. Your muscle cells have to stay muscle cells. There's also a risk of cancer if you take cells back to pluripotency. They could divide uncontrollably. So this approach would not work inside your body. But the discovery that came out of the Sinclair lab at Harvard that I think is incredibly profound is that if you treat with just 3 of the 4 factors, OSK, what they observed experimentally was they did not get pluripotency, So there was no nanog expression, there was no increase in tumors. In fact, there was a slight decrease in tumors, and we can talk about why that would be. And yet they had profound effects at the cell, tissue, and organism level, so much so that it was put on the cover of the journal by the editor. The editor's the one who decide, right, what goes on the cover. They're also the ones who decide the title, Turning Back Time, and this image, which is an eye with a clock going in reverse. Because what they demonstrated in this study is that they took mice that were blind, whether from glaucoma, a model of glaucoma where they insert beads to cause it, whether from age, or whether from physically crushing the optic nerve, and restored their vision all the way back, not just to the vision of an old mouse, but the vision of a young mouse. That's incredible, right? Never been done before. Another way to see this and understand what's so profound about it is just think about the central dogma that we all learned hopefully in middle school, right? So DNA, RNA, proteins to metabolites, and think about what's different and what's the same across two different cell types, so nerve versus muscle, or two different cell ages, old versus young. So you can look at two different cell types and distinguish them just by their metabolites. Same with cell ages. You can distinguish them just by the proteins they have, by what RNA is active, but they have the same DNA. And so it doesn't take kind of a rocket scientist to look at this diagram and say, well, what is it that's happening here that might be changing the outcome for these two different cells? And the thing that happens there is epigenetics. It's now established wisdom that the thing that determines cell type is epigenetic. So the thing that tells it to be a nerve cell or a muscle cell is the epigenetic. I believe it will become established wisdom that the upstream thing that is causing cell age versus young will ultimately be demonstrated to be the epigenetic code. It's not that there's a lot of other downstream hallmarks. There are. Lots of other things change, including you get some mutations as areas get hypomethylated. But if you want to think about manipulating and actually taking something all the way back, rejuvenation, I think this is the only way. So given time, I'll go quickly over this. Maybe I'll skip this one. Okay, so we've created a company to make this real, bring it into clinical practice. We're starting with, since the original work was done in the eye, we're starting with optic neuropathies, glaucoma, and NION. These are serious diseases that affect a lot of people. Number one cause of blindness in the elderly on the left, number one acute cause of blindness on the right. We have now finished preclinical work. So the original work was done in mice. We've now recapitulated it in non-human primates, NHPs. And we see an incredible logic flow across the board of everything you would want to see in these preclinical experiments. So we know that we're getting the OSK into the tissues and it's being expressed. That's the brown dots. We know that we're taking that methylation, the epigenetic pattern, right back to youth. That's the yellow-blue back to the original yellow-blue pattern. We know that we're improving cellular function. So these are nerve cells, so they have an electrical impulse. As you go blind, they get attenuated. The period gets lengthened. And then after treatment, we see this incredible recovery of the shape of that curve. In the fourth column, those first three were all from primates. This fourth image is actually from mice from the original paper, showing that's an optic nerve that's been crushed. You can see the axons are dead. And then after treatment, the axons are literally regrowing back down the optic nerve, never been seen before. Able to test vision directly in mice. We unable to directly test it in NHPs, but we believe, given everything else we saw, that it will also translate. And I'm very happy to announce that we've now finished our what's called our GLP tox, good laboratory practice toxicology. In NHPs, there were 0 significant adverse events. So we're moving forward into clinical trials in humans starting in Q1 of next year. We will be dosing patients in our phase 1. It's designed for safety and tolerability, but we should have anecdotal efficacy data even by the end of next year, which obviously is going to be very important for us as a company, but I think it's going to be a blockbuster moment for the field if it works, obviously, because it will be the first time we've actually shown a rejuvenation event at a cellular level in a human. It's not just the eye. There's a ton of data out there to show that this affects all organs and systems. These are all different labs showing that this applies everywhere. We wanted to demonstrate that with a little bit of data. So we're working on a few other organs we haven't released, but we have released data from liver. Same drug. Like, there's no reason in the world, if you come at this with a traditional medical paradigm, that a glaucoma drug should work on fatty liver disease. But that's exactly what we see. If you come out from a rejuvenation viewpoint, it becomes easier to understand. So we see all these parameters improved without affecting body weight, because this is not a body weight drug. This is a drug that we believe is rejuvenating the hepatocytes. So I'm going to stop there. I think 3 big takeaways. As I look ahead, I'm just incredibly optimistic for not just what we can do, but what as a field we can do to affect personal health. Without commenting on the exact size of the bars, right? I believe we can look forward to longer lifespan and shorter period of ill health. There's tremendous economic value here, and I believe it's as profound, more profound than as some of these other things that we now take for granted that used to seem like science fiction, right? Whether it's flight or computers or the internet or now AI. So very happy to connect afterwards with anyone who would like to. The QR code is there. Thank you.
Michael, thank you. Okay, maybe just 1 or 2 quick questions I think we have here. Are there specific age-related diseases where partial reprogramming appears most promising?
And actually the second question, I'll take them both together.
Well, you could, take the second.
Yeah.
So I'll just
I'll
read
take them
out.
both together, actually.
Okay, cool.
Yeah.
So the second question is there a pathway to deliver cellular reprogramming to the whole organism?
Yeah. For our therapy to work, you have to get 3 proteins, little transcription factors, very small proteins, inside a cell. But that's hard to do. We don't actually have technology that delivers proteins inside a cell easily. There's 3 kind of basic ways. One is you can deliver a DNA plasmid in a vector, and then the DNA will then make RNA and then the protein. And there's various vectors, AAVs, lentivirus, et cetera. The second main way is, which we're all familiar with now, is you can deliver mRNA in a lipid nanoparticle, which like the COVID vaccine, or some of COVID vaccines. And then the third is, could you actually get the protein directly in with an exosome or something like that? The delivery technology is still emerging in this area. And so on the first question, second question, could you get this into the whole organism? Yes and no. There are vectors that deliver everywhere-ish, but they deliver more in certain organs than in others. And that's a problem, right? Because you kind of want equal dosing. So there's not a perfect technology yet for this. And then there's some cell types that get missed. So the other question is, are there certain diseases that this is better for? Well, we think it works in all age-related diseases, but given the hardest thing to do is delivery, getting it into the right organ, what we're prioritizing for now is what are the areas where it's easier for us to deliver? And there are certain organs where that is easier. The eye is one of them, the liver is another. There are others, which I'm not disclosing, but that's how we're thinking about it right now. But our hope is the field is working on delivery well outside just our little company, and that this will get easier and easier over time to get proteins into the cell, and we can think about knocking off age-related disease after age-related disease in sequence, hopefully, and then maybe one day, hopefully, whole body rejuvenation. It's a long way away. It may not be even in time for us in the room.
How long?
But yeah. Maybe longer than us in the room.
Could you hurry up?
But certainly for our kids, I think.
Okay, Michael, thank you very much. Thank you.